Okay, so now that we've considered the chemical bonds and some of the smaller molecules like water that are important to living organisms, we're really ready to turn our attention to the large molecules that are found in living tissues. I do want to point out, however, that if you look at the graph on the left-hand side of your screen, you'll see that by mass anyways, the largest proportion of any compound that's found in living tissues is actually water. That's what's represented by this 70% in blue here. So what we're going to be focusing on now for the rest of Chapter 2 is the large molecules which make up living organisms and the chemical reactions and some of the laws of thermodynamics that govern their synthesis. Now, of the macromolecules, proteins make up the largest percentage of the mass, followed by nucleic acids, carbohydrates, and lipids. And I'm going to use the term macromolecule, and I just want to be clear about how I'm using that term, and that is to describe a polymer that weighs more than 1,000 Daltons. Now, all biological macromolecules share something that's called biochemical unity, which simply means that they're found in all living organisms. These biological macromolecules are all organic, meaning that they contain carbon, and they're only found in living organisms. In all cases, their structure and function are related, and we're going to say a lot about that in this upcoming 2 chapters, actually. And with the exception of the lipids, they all have in common that their single units, or monomers, are held together by covalent bonds, which, if you remember from the last part of this video, are very strong bonds that are formed by the sharing of electrons. So the functions of these building blocks are many and varied, and I'm not going to go through this list now because as we cover each of the macromolecules, I'll mention some of these. All I want you to see right now is that they really do perform all of the functions that make life possible. So a couple of terms to clear up that I'm going to be using. The first of which is the term monomer, and a monomer just refers to a single unit of a compound. A polymer, with poly meaning many, consists of many monomers that are linked together with covalent bonds. And as I mentioned before, the term macromolecule refers to a very large polymer that weighs over 1,000 Dalton. So if you look down on the bottom of the screen down here, I have an example for you of a polymer. This is a polymer which is known as starch, and it actually consists of glucose units, which are the monomers that make up the polymer. So in this case, this is a monomer, and here is the covalent bond that joins it to the next glucose in the chain. You can sort of think of this as something like a train, a passenger train, where each of the cars would be a monomer and the entire train would correspond to a polymer. So, just to get you familiar with what some of these biological molecules are and their associated monomers, I've included this table from your textbook. And this shows us that the monomer that is found in proteins is the amino acid. For carbohydrates, which are also known as polysaccharides, the monomers are monosaccharide sugars. For nucleic acids, the monomers are nucleotides. So, there's a very specific chemical reaction that is involved in forming these polymers, and this is what's referred to as a condensation reaction. Sometimes you'll hear these referred to as dehydration reactions, and the reason for that is that a molecule of water is released every time a covalent bond is formed between these two monomers. So for example, here you have 2 monomers, and after this reaction, there is the release of a water molecule, and now you have a monomer joined to another monomer, which we would call a dimer. I also want to point out that I've added something to this slide which wasn't in your book, and that is this condensation reaction actually requires the input of energy. And we'll talk some more about that later, but I just wanted to get that up on your radar now. Now, in the case of condensation reactions, there is 1 molecule of water that's removed for each covalent bond. And in the cell, this is actually aided by enzymes, which we discussed in lab. I also want to introduce you to another term, and that is an anabolic reaction. So whenever you are building chemical bonds, this would be called an anabolic reaction. This is easy for most athletes to keep straight because most people because athletes who choose to use anabolic steroids are doing so because they want to build muscle mass. So condensation reactions are also anabolic, meaning they are building a more complex compound from more simple compounds. The body is also constantly breaking down complex molecules. For example, in the proteins in the meat that you might eat, it has to be broken down in your digestive system into its constituent amino acids. The reactions that involve the breaking up of a polymer are actually called hydrolysis reactions, and they get their name hydro from water and lysis meaning to split, and it's basically the reverse of the condensation reaction. So here we have a trimer, which consists of 3 monomers, which are joined together by 2 covalent bonds, which are shown by the red bars here. And every time a water molecule is added, one of the monomers is split off. So this slide is actually showing 2 successive hydrolysis steps. And again, I wanted to we'll come back to this concept of energy, but I wanted to introduce the idea that as chemical bonds are broken, energy is actually released in this kind of reaction. And this is what we would refer to as a catabolic reaction. Also, I want to switch back to the condensation reaction for just a moment and introduce one more term to you. The term that we use to describe a reaction which requires energy input is an endergonic reaction. Conversely, a reaction in which energy is released is called an exergonic reaction. Alright, so we're ready to turn our attention to the first of these biological macromolecules. And first we're going to consider the carbohydrates. Carbohydrates get their name because they contain carbon, hydrogen, and oxygen, and they contain those in a ratio of 1 to 2 to 1. So the carbo comes from the carbon, and the hydro comes from the hydrogen and oxygen that are found in carbohydrates. These function in the cell as an energy source and also as a carbon skeleton, meaning that they are modified into many other compounds. Now, I just want to be clear that this ratio I described to you, it only occurs actually in the monomers. And many of you are probably familiar with carbohydrates because for a long time we were told they should make up a pretty good portion of our diet and form the basis of the food pyramid. So one of the most important carbohydrates is glucose. The chemical formula for glucose is C6H12O6. If you know anyone who is a diabetic, as this slide is showing down here, someone getting their finger pricked, you know that they have to follow very closely their blood sugar levels, and the sugar they're actually testing for is actually glucose. So glucose is extremely important to all animals. It is readily used as a food source, and in fact it is the only sugar that can be metabolized by the human brain. So glucose can either exist in a straight chain form or in a ringed form. In cells, it usually exists in the ring form. And there are a couple of different variations on that structure that depend upon the orientation of 2 atoms on the number 1 carbon. If the hydrogen atom is sticking straight up on the first carbon, then that's called alpha glucose. And if the hydrogen atom is pointing the opposite direction, it's called beta glucose. So, in terms of forming polymers of carbohydrates, which we call polysaccharides, it is often glucose monomers that are involved. Think for a minute about what kind of reaction this would be. Here is a water molecule being released. The blue boxes over here are showing where that's coming from off those 2 monomers. And in this case, we are seeing the formation of a compound called maltose from alpha glucose and a beta glucose. Again, in the cell, this would actually be catalyzed by an enzyme, one specific enzyme. So ask yourself, is this a condensation reaction or a hydrolysis reaction? Is it anabolic or catabolic? And what would you predict about the energy requirement for this reaction? Polysaccharides are formed in the method we just described. They are very large polymers of monosaccharides, and they are connected by a special kind of covalent bond which is called a glycosidic linkage. They vary in their degree of branching, and this degree of branching has a lot of bearing upon their function in the cell. And remember, I did say that the structure and function of biological molecules are related. So let's take a look at this. But first, let me show you what I mean by branching. So in the top polymer that's shown here, this is a very straight or linear molecule. It would not have a lot of branching. And this is the compound known as cellulose. By comparison, another polysaccharide, starch, and also glycogen tend to be much more branched than cellulose. And so by branching, we mean that we're referring to this molecule that's sticking off right here. So this is a diagram just to give you an idea of what we mean by the branching structure and how this sort of affects the function. So the first example we looked at on the other screen was of cellulose, and we said that cellulose was very linear. How this impacts the function is it means that cellulose molecules will pack together very, very tightly. And for this reason, it means that cellulose is a very important compound that provides structural support, particularly to a lot of plants. It is a major component of plant cell walls and also the most abundant organic compound on Earth. Its structure is very much in contrast to that of starch, which you see in the middle of the screen here, which is quite branched. And the function of starch is as an energy storage, mostly for plants, entirely for plants. In contrast, glycogen, which is energy storage for animals, is shown on the right-hand side, and this is a highly branched structure. So this diagram is actually just showing how cellulose fibrils pack together, and you can see the linear cellulose strands here, and I just want to call your attention to all the dashed lines. Do those dashed lines look like anything you might have seen in the last online lecture? You might want to go back and look at the diagram on hydrogen bonding. And that's what you were thinking, you're exactly right. So all of these dashed lines indicate hydrogen bonds. So one of the things that makes cellulose so stable is that it is held together by many, many, many hydrogen bonds. So one of the things I want you to keep in mind about carbohydrates, that these are exactly the kinds of food sources that you're going to be providing to the yeast in your fermentation experiment. All right, the next group that we're going to discuss are the lipids. And the lipids are very interesting. They are a very chemically diverse group. And what's interesting about them is that just about the only characteristic that unites them is the fact that they are all hydrocarbons, meaning they contain lots of carbon and hydrogen atoms that are insoluble in water. And so as soon as you hear that, you should be thinking these are nonpolar covalent bonds. Those examples of some lipids that you might be familiar with include walnut oil, olive oil, and butter. So the first category of lipids that we're going to consider are the fats and the oils. Their function is in energy storage. And this is a slide showing you an example of a particular kind of fat that's called a triglyceride. And a triglyceride consists of a glycerol molecule that is joined to 3 fatty acid molecules. Look, here are 3 water molecules being released in the formation of this compound. Is this a hydrolysis reaction or a condensation reaction? What are the energy requirements that you would predict for this reaction? There's a special name for the covalent bonds that hold these fatty acids to the glycerols, and these are called ester linkages. And unlike the other biological molecules we're going to talk about, this name is not unique to a type of bond that's found in lipids. The glycosidic bonds that we discussed that were formed in the carbohydrates are unique to carbohydrates. And when we get to proteins and nucleic acids, you'll see they have a special kind of covalent bond that's found only in those macromolecules. So the lipids are the only one that do not have a covalent bond that's found only in lipids. There are 2 distinct categories of fatty acids that are divided based upon the structure of the fatty acid tail. And these are the saturated and the unsaturated fatty acids. And on the left-hand side of the screen, you can see palmitic acid, which is an example of a saturated fatty acid. It's called saturated because, if you'll notice, there are only single bonds between the carbons, which mean that every single carbon in that tail has bound to as many hydrogen atoms as it possibly can, and still be bound to 2 other carbon atoms. We call that saturated. On the right-hand side of your screen, you can see linoleic acid, and this is an example of an unsaturated fatty acid, because you'll notice in the areas that are highlighted in yellow that there are 2 places where there is a carbon-carbon double bond. In that case, those particular carbons are not saturated with hydrogen atoms. Now, these differences in structure have very important implications in some chemical properties of these 2 fatty acids. The biggest one is that the saturated fatty acids, like palmitic acid, because they are so straight, they can pack together very, very tightly. Those tend to be a solid at room temperature. In contrast, the unsaturated fatty acids, like linoleic acid, because of that kink in the tail, they are actually a liquid at room temperature. So the arrangement of the chemical bonds in these tails of the fatty acids are extremely important. And they're not just important because they determine whether something is a solid or a liquid at room temperature, but they're also very important in terms of your body's ability to metabolize them. I suspect that many of you have heard of trans fats. On the left-hand side of your screen over here is a fatty acid which is called iliadic acid. This is a great example of a trans fat, and it's called trans because of where this double bond is occurring here. This is a different arrangement, which we call an isomer, of the compound that's shown over here on the right. This is oleic acid, which has the cis arrangement, CIS, cis arrangement of this double bond, as compared to the trans arrangement over here. And this is actually found in olive oil. These make a very big difference to your body because your body is able to metabolize the oleic acid, but is not able to metabolize the iliadic acid. So this is one of the reasons why we're told not to eat trans fats. Your body actually cannot metabolize them, and in fact the trans fats will just accumulate in your body. Some of it is excreted in waste, but much of it accumulates in your vascular system. So you might be wondering, where do these trans fats come from in the first place? And an interesting historical fact is that they were actually created to try to make things cheaper for the restaurant industry. In particular, the goal that they sought was to try to find a frying oil that did not spoil. So, this is one of my soapbox moments. I don't have very many, but you really should try to stay away from trans fats. And you should read the labels very carefully because you may not be aware of this, but in the United States, the food label can be labeled 0 trans fats as long as there is less than 0.5 grams of trans fats per serving in the food. The really scary thing about that is that no amount of trans fat is recommended for us. 0. You can't digest it. And it's known that 2 grams of fat per day can cause all sorts of problems. So, how do you get around this? Well, you actually have to read the label and see if it contains hydrogenated oils. And if it does, you know that you're eating something with trans fats in it, and you should probably make a healthier choice. There's another very important group of lipids that I want to mention, and these are the phospholipids. And phospholipids have a very important function because they actually form biological membranes. Phospholipids consist of 2 fatty acids, which are shown here, plus a phosphate group, plus a glycerol. So this is the phosphate group, here's the glycerol, and here are the fatty acid tails. This creates some interesting characteristics for this molecule because you basically end up with a charged region where the phosphate group is and an uncharged region where the hydrophobic tails of the fatty acids are. So oftentimes you'll see a phospholipid drawn in this way where it has a hydrophilic head and a hydrophobic tail. Just as we saw in the triglyceride, these fatty acid tails are also joined to the glycerol through ester bonds. So it's this amphipathic quality of phospholipids that makes them so readily able to form membranes. If you take phospholipids and you put them in water, they will spontaneously arrange into a bilayer. So it's a layer of two different rows of phospholipids, and they arrange themselves so that their hydrophilic heads are pointing out and in contact with the water, and the hydrophobic tails are pointing to the inside. They'll do this spontaneously. So this arrangement is the same one that we see in cell membranes, and it's actually energetically quite favorable. It helps keep the watery contents of the cell inside. It prevents water from outside leaking into the cell. And it forms a way to compartmentalize the cell. So you'll hear this arrangement referred to as the phospholipid bilayer. Steroids are also an important class of lipids. They do not really have a standard structure, but they're described as a chemical linking of something called an isoprene unit, which has 5 carbons and 8 hydrogens. I just like to call it, they're sort of multiple rings that share carbon atoms. And they have very different structures from phospholipids and also from each other. For example, cholesterol forms the backbone for many hormones. So even though you're told to limit the amount in your bloodstream, we actually do need some cholesterol. It's also an important component of cell membranes. Cortisol, for example, is the stress hormone. It is released in response to stress, causes an increase in blood sugar, suppresses the immune system, and aids in fat and protein metabolism as well. And finally, testosterone is responsible for the development of secondary sex characteristics in vertebrates. So, as you can see, these steroids have quite diverse functions. There's even one that's actually a vitamin. And then the last group of lipids that we're going to consider are the waxes, and these groups have in common the fact that they are highly nonpolar. They have this characteristic because they have extremely long and very saturated fatty acid tails that are joined by ester linkages. Waxes can be found on the feathers of birds. They can prevent the birds from getting too wet to fly, and also on the waxy cuticles of desert plants.